Dear students, I, Mr. Vishal Shah, Assistant Professor, Krishna Institute of Pharmacy, Karar, once again welcome you all to this another session of the fats and oils. In this video, we are going to discuss about the remaining classes of the compound lipids. In previous video, we have discussed about the two phospholipids classes, phosphoinosirates and phosphosphingosides. Also, we have discussed about the glycolipids. In that, we have covered one part, that is the cerebrosides. In this video, we are going to discuss about the second class of glycolipids, that is the gangliosides, as well as aminolipids, sulfolipids. These are the various few classes of the compound lipids, also lipoproteins. So let's begin with our today's session. Before that, just we will see what are the learning outcomes of this today's session. After this session, students will able to explain the classification of the compound lipids. Now, first class that today we are going to discuss is gangliosides. As I said, this is the class of the glycolipids. Now, in 1955, Klein, that is the name of the scientist who has isolated a new type of glycolipids from the brain tissue and named it as a ganglioside. These are found in significant concentrations in ganglionic cells of nervous tissue. That's why they are known as the gangliosides, and also in most parenchymatous tissues like spleen and erythrocytes. They are make up about the 6% of the membrane lipids in the grain matter of the brain. They are also found in lesser amounts in the membranes of most non-neural tissues. Gangliosides are thought to act as a receptors for the toxic agents like pathogens, Vibrio cholerae, influenza virus, as well as tetanus toxin. They are also implicated to play a role in cell-cell interaction. The structure of gangliosides is very complex and it is related to cerebrosides. In that, they contain a ceramide, which is nothing but the N-acyl sphingosine, linked to a carbohydrate, that is the galactose or the glucose. Now, the cerebrosides or glycosphingosides are there. They are mainly consisting of the sphingosine molecule. Here, instead of the sphingosine, they are mainly consisting of the ceramide, which is the N-acyl sphingosine, which is linked to the carbohydrate, that is the galactose or the glucose. So this is the basic difference between the cerebrosides and gangliosides. Means cerebrosides are consisting of sphingosine molecule, and in case of gangliosides there is a presence of N-acyl sphingosine. That is the nitrogen atom of the sphingosine will be acylated. So here we have finished with the gangliosides. In this way we have finished with the glycolipids. Glycolipids are nothing but these are the lipids in which there is a presence of the sugar moiety as a non-lipid portion. They are having two types. One is the cerebroside and other one is the ganglioside. Now, next class of the lipids or compound lipids is aminolipids. Phosphatidyl ethanolamines, then serines are aminolipids, and sphingomyelins and gangliosides contain substituted amino group. Aminolipids means what? Those lipids which are consisting of amino functional group, they are known as the aminolipids. As you can see, these various names are repeated over here because all these compounds are consisting of the amino functional group. So due to the presence of amino functional group, they are also known as the aminolipids. Phosphatidylcholine, ethanolamines, then serines, all these amino acids or aminolipids these lipids are consisting of the amino functional group. That's why they are known as the amino lipids. Then after this, next class of the compound lipids is sulfolipids. They have been isolated from the brain and other animal tissues. These are the sulfate derivatives of galactosyl residues in the cerebrocyte. Means whatever the galactosyl residue that is present in the cerebrocyte cell, their sulfate derivatives are known as the sulfolipids. So this is the common structure of sulfolipids, where you can see again a molecule of glycerol. Here you can see the molecule of glycerol that is present, in which first carbon atom is again linked to the free fatty acid, second carbon atom is linked to the again free fatty acid. Probably the first free fatty acid is the saturated free fatty acid, and the second free fatty acid that is present is unsaturated free fatty acid. Now in addition to that, there is a presence of the galactose unit, and this galactose unit that is present, in which this particular residue is having the sulfate derivative or sulfur group, you can say. That's why these types of the lipids are known as the sulfolipids. So this is about the sulfolipids, that is the fourth class of the compound lipid. Then now last class of the compound lipids is lipoproteins. Lipoproteins are hydrophilic complex containing triacyl glycerol, phospholipids, cholesterol, cholesterol esters, free fatty acids, and the proteins. These are the compound lipids found in plasma and the cell membrane. These lipids are synthesized in the liver as well as in the adipose tissue. They are mainly insoluble in water, hence difficult to transport from one part to the another part of the body. Lipoprotein A, in combination with other lipids and proteins, are water soluble, and they are significant for the transportation of the lipids. Pure fat is less denser than water, so as the proportion of lipid to protein in lipoprotein increases, hence density decreases. Means if the proportion of lipid is more into that particular protein, then the density decreases. So depending upon the physiological function, lipoproteins can be categorized into 4 categories. Also depending upon density also you can say that they are classified into 4 categories. They are namely chylomicrons, then LDL, which is known as the low density lipoprotein, then VLDL, that is the very low density lipoprotein, and HDL, that is the high density lipoprotein. One by one we will discuss about these lipids or lipoproteins. First we will discuss about the chylomicrons. These are the largest and least dense of the lipoproteins, a cluster of lipids and proteins that are used as a transport vehicle for the fats. So these are the least dense because they are consisting of more proportion of lipids as compared to the proteins, and that's why they are used as a transport for vehicles or the fats. Chylomicrons transport diet derived lipids, that is the triglycerides, from the small intestine through the lymphatic vessel and into the bloodstream and rest of the body. Means major function that is served by the chylomicrons is transportation of diet derived lipids, that is the triglycerides. We have studied in detail about the triglycerides. They are consisting of the esters of glycerol with the free fatty acid. So they are transported from small intestine to the other parts of the body through the blood stream. As chylomicrons pass through the blood stream, body cells removes the lipid from them, and the liver cell removes the remaining portion of the chylomicrons from the blood and reassemble then into the new triglycerides. And due to the removal of lipids from the chylomicrons, their density increases and they will become more denser as compared to the chylomicrons. Next type of lipoprotein is VLDL. That VLDL is nothing but the very low density lipoprotein. Very low density lipoprotein are made up in the liver from the chylomicrons, and they are transported mainly triglycerides. About 50% of triglyceride is transported by using very low density lipoprotein, some cholesterol and the phospholipid molecules. The VLDL, that is the very low density lipoprotein, travel through the body and cells remove the triglyceride from them. As they lose triglyceride, the proportion of cholesterol increases and they, as become more dense, they become very low density lipoprotein. Means after losing triglycerides from the VLDL, the proportion of the cholesterol increases and they will be converted into more denser, and they are known as the LDL. Means VLDL will be broken down into the LDL, that is the low density lipoprotein. Low density lipoprotein are composed of primary of the cholesterol. About 50% of the cholesterol is present in the LDL. They circulate through the body and release the triglyceride, cholesterol and the phospholipid into the body cells. Now we know that LDL is associated with the very famous disease, that is the atherosclerosis. This is the condition in which there is a deposition of the cholesterol inside the blood vessel, and due to which narrowing of blood vessels takes place. So this is the one of the reason for the heart attack or the ischemic heart disease. So why? Because these lipoproteins mainly consisting of the cholesterol, and when they are hydrolyzed, they will be converted into the triglyceride, cholesterol and phospholipids. So these cholesterols are deposited on the surface of the inside wall of the blood vessels. Now body cells collect the lipids and use them to make the cell membranes, hormones, or store them for the later use. In liver, LDL will be removed from the circulation. This lipoprotein represents the final stage in the catabolism of the VLDL. Now next and the last lipoprotein is high density lipoprotein, which is also known as the good cholesterol. High density lipoprotein transports cholesterols from the cells back to the liver for a recycling or disposal. Means here you can see the difference. LDL will carry out the transportation of cholesterol from the liver to the rest of the organs, or from small intestine to the rest of the organs, and the HDL will take the whatever the high amount of cholesterol that is present from the cells back to the liver, and there it will be catabolized. So these are involved in the VLDL, LDL and cholesterol metabolism. That's why HDL is known as the very good or good cholesterol. They are involved in the transportation of the cholesterol. Now the functions that are served by the HDL, they transport and derive the lipids to the tissues to maintain the structural integrity of cell surface and subcellular particles like mitochondria and microsomes. They are required. So this is about the classification of compound lipids. The beta lipoprotein fraction increases severe diabetes mellitus. Now you have exercise. Go to this particular website. Use the password that will be provided to you in your group and answer the questions that are given on this particular website. Thank you. If you have any queries related to the pharmaceutical organic chemistry, you can contact me on this mail. If you like the content of this video, you can subscribe to my YouTube channel and share with your friends. Thank you once again.